Hello everyone, welcome to video 1 of the Paper 6 Tips and Tricks series, where I will be telling you guys everything you should know about the Paper 6 and how to solve each type of question and the basic stuff you should know about it. And this video will also help you if you're taking other subjects as well in chemistry, physics, Paper 6 as well. So this is just the basic information you should know in paper 6 generally. So some of the stuff will be biology related and some of the stuff will be just general knowledge about the maybe independent variable, dependent variable, stuff like that. So this is the plan that I will be doing. I will be doing 5 videos where I'll be covering every single thing you should know about the paper 6. This is video 1. I'll be explaining today the designing an experiment, where I'll be covering the following things today. I will be telling you guys about the safety and improvements to the experiments. If you're solving past papers, you will see questions that are like suggest an improvement to this method, or maybe they're asking about safety measurements we should take, or they're asking about safety equipment. So also I will be explaining the food test, such as Benedict's, iodine, DCPIP, and so on. I'll be also explaining how to design an experiment, like how to write it down and what to write exactly, and how you will be awarded for your question in the exam. And I'll be explaining the dependent, independent, and constant variables. So if you want to know about everything, so please keep on watching. Now, to begin with, you have to first identify the independent and dependent variable from the sentence or the passage that he gave you in the experiment in the question. So first, let's just keep in mind that the independent variable is the something that you keep on changing, or you're going to take many values of in order to exactly know like how will it affect the maybe the dependent variable. The dependent variable is the something that you are measuring, or the something that you're seeing the effect of the independent variable on. Like, for example, I'm going to give you an example now. The effect of amylase, the pH on amylase activity. Amylase is an enzyme that breaks down starch into maltose, and then maltase breaks down the maltose into simple sugars, but we're going to now focus on the amylase for now. So, over here, we can know two things. We can know the effect of pH, and the pH over here, we can know that this is the independent variable, which I just said is the something that you keep on changing, and the thing that you can take many values of in order to see its effect on the dependent variable, which is the something that you are measuring, or you're seeing the effect of the independent variable on. So over here, how do we identify the independent and dependent variable in the sentence over here? Actually, whatever, but this is not going like most of the time it's like that, like whatever comes after the effect of in the sentence, then it's the independent. But sometimes he doesn't say the effect of, but you should understand what's going on. But this is like just a tip for you. Whatever comes after the word the effect of is most probably the independent variable. You can keep that in mind. So over here, the effect of pH, which is the independent variable, we're going to use at least 3 values of the pH. This is like the minimum number. You can add more if you want, but this is the minimum number just to be in the safe side. For example, we're going to use pH 7, pH 4, pH, let's say 5. So this is something you're going to change, and you're going to like just see how will it affect the amylase activity, which is the dependent variable. But I have to tell you something about this. In the exam, if he asks you what is the dependent, you can't just write down the amylase activity. You have to write down either the time taken for the starch to disappear, or the time taken for the amylase to produce maltose. So you can't write down dependent variable as just like that, the amylase activity. No, you have to write it down as something you can physically measure in a lab, something that you can see, or something you can, like, you know, in the lab. So in the lab, if you go and you're trying to measure the amylase activity, what exactly are you doing? What exactly are you going to, like, measure? Like, still, you need to do some extra measurements, extra procedures to know what is exactly are you doing in order to know the amylase activity. So this is what's meant by the dependent variable. It is something that you are measuring, and this thing that you're measuring is going, of course, the independent is going to have an effect on it whenever you change it. So we all know that the amylase optimum pH is pH 7. And if we change it any further, it's going to denature, and the active site is going to change, and it will no longer fit in its substrate. So this is what's really like an idea of what's the independent and dependent variables. So again, the independent variable is the variable that you are changing, and you're trying to investigate what will happen if you change it. And on the dependent variable, and the dependent variable is the thing that you are measuring physically in the lab, and the thing that you are trying to see what will happen to it whenever you're changing the independent variable. So that's basically just like the independent and dependent variables. Now moving on to the constant. This is something also you should keep in mind while you are doing an experiment. Constant variables is something that you are keeping the same throughout the investigation, and if you change it, the experiment might be inaccurate. So, for example, in this investigation, we should, of course, keep the amylase concentration constant. What do I mean? You should keep this thing like the amylase volume, you should keep it the same in every pH over here. For example, in pH 7, let's just say he's going to use 3 centimeter cube of amylase in each of the test tubes in containing different pH. So you can't just like say, oh, in this pH 7 I'm going to use 3 centimeter cubed of amylase, and in pH 4 I'm going to use 4 centimeter cubed, for example. No, this is something, if you're going to change it, the experiment will not be accurate, and it's not going to be like, fair. Like one pH has 4 centimeter cubed and one pH has like 5 centimeter cubed. This can't be correct. Each pH should have the same amount of amylase. So this is what's meant by constant variables. So this is just an example, and you should keep this thing constant. This is what's really meant by the constant variables in the experiment. Now, moving on to the control variable. The control variable is something that you are basically going to, like, for example, in this experiment, you're going to use maybe water to know, like, if really the amylase or the dependent variable is causing the change in this experiment. Like, the control variable is like the something that you are going to use, in which you are going to prove that yes, the amylase is doing the difference in this experiment. The amylase is the thing that really made the difference and really changed the time of the uh, starch or whatever you're going to use. And yeah, so for this experiment, maybe we're going to use water instead of amylase and see the effect of pH, or you're going to use water and vice versa. So this is what's really meant by the control variables. Something that you are going to use instead of the dependent, in order to prove that the dependent variable is really causing the change over here. So this is just an idea of the dependent, independent, constant control variables. So of course, when you are writing down the experiment in the exam, you should include the dependent, constant, independent variables as well. Well, sometimes you don't need to write down the control, like sometimes, but it's preferred if you do so. Like in this experiment, you can write down, you can use water as a controlled variable, but that's not always needed. But what's really needed is the dependent, independent, and constant variables. These should always be present in your experiment. Now, moving on to food tests. This is something you should already know from chapter 4, biological molecules, but we're going to say it again. Now, we have 5 substances that you should be able to do food tests for, which is reducing sugars, protein, fat, vitamin C, and finally starch. I'm going to explain how to test for the presence of each of the substance that I just wrote right now, and how will it look like if it's a positive, or how will it look like if it's a negative. Now, first we're going to discuss the reducing sugars. And before I start, I just wanted to point out that sucrose is not a reducing sugar. And you will know more in chemistry, but sucrose is the only non-reducing sugar. Like, it's not a reducing sugar. So if you test using Benedict's with this substance, it will not work because it's not a reducing sugar, and this test is for reducing sugars. Now, first of all, you should use Benedict's reagent for the reducing sugars. And sometimes in the exams, they get a question and they tell you, how will you test for reducing sugars in a specific substance? Or if it's part of another question, they're going to tell you how to, for example, test for reducing sugars in whatever thing. So this is what you should write. You should write in the question Benedict's reagent. This will give you 1 mark. So this is the substance that you're going to use to test for the reducing sugar. And you should also do another step. You should heat at temperature 80. Now this step is crucial. It's very, very important. You should do this step using a thermostatically controlled water bath. I'll write it down, just give me 1 second. Now, it's very important to write thermostatically controlled water bath. Why? That's because thermostatically controlled water bath is like a device that is used in labs, that it helps to maintain the temperature at a specific value. For example, if we're going to use the thermostatically controlled water bath, we are going to set it to temperature 80, and this is where the temperature will stay constant and will not change, unlike the normal water bath that we have seen in various experiments in the past papers. So you should write down thermostatically controlled water bath. So what exactly are you going to write if the question is 3 marks? Now, this thing is very important and you should know it. This is the positive result and the negative result. Now, what will happen if you test the substance and you heat it at temperature 80 using the thermostatically controlled water bath? What are you supposed to see to indicate that there is reducing sugars or not? And how will you know if there is reducing sugars or not using this test? Now, for the positive result, we have several colors. We have red, brick red, orange, yellow, and green. Now, these colors can indicate that there is some reducing sugars present. Now, if you're going to go in order, red is, if there is red color that appeared, that means there is very much reducing sugars, or there's a high quantity of reducing sugars in this substance. Now, if you move again, orange means that there is a lower quantity than the red. Yellow means that there is less quantity, and so on. Green is like I think the least. So it won't matter. You can just write red, and all of these are correct. but you should write down red. This means there is very a high quantity of reducing sugars. Now the negative result is going to be blue. That's because the Benedict's reagent is actually blue in color. And if you're going, 1 second, I'm going to get you a picture of the thermostatically controlled water bath and also a picture of the Benedict's reagent alone, like in a test tube. 1 second. Now this image shows the Benedict's test results if you're going to do it in a lab in real life. Now the blue solution shows a negative result. This shows that there is no reducing sugars present in the substance or whatever you're testing. So if you get this color after doing the experiment, then there is no reducing sugars in this substance. Maybe there is another substance that we will be talking about in this video later. Now, green, orange, brick red, these all show that there is reducing sugars present in this substance. So if you get any of these colors, then there is reducing sugars in your substance. So this is the Benedict's reagent test and how to test for reducing sugars. And this is an image of the thermostatically controlled water bath that I was talking about. This thing, you can use it and put it, and you can set the temperature over here in this thing that you see over here. And you're going to set it down, and you will set it down if you're going to do Benedict's reagent test. You're going to set it down to 80 in order to the Benedict's reagent to be effective. Now that was for our reducing sugars. Now moving on to the protein, it's much shorter than that, so don't worry. The protein, we're going to use something called the Biuret reagent. This is the substance that we'll be using to show or indicate that there is some protein in our substance. So there is no thermostatically controlled water bath or heating required in any of the next food tests that I'm going to talk about. It was just the reducing sugars that requires heating and requires the thermostatically controlled water bath. So just point out that only required in the reducing sugars test so now the Biuret reagent, as we said now, it's for the protein. Now what will happen, you're just going to put down the Biuret reagent into whatever you're testing, and if you get a purple color or lilac or violet or whatever you call it, purple color, then indeed there is protein present. If you get a blue color and the Biuret shares or has something in common with the Benedict's, it's actually blue in color, just like the Benedict's solution. So if you still do the experiment and you get the blue color, that means it is a negative result and there is no protein in this substance. So that's it. You're just going to use Biuret reagent, and if you get a purple color or a violet color, then protein is in this substance. But if you get a blue color, then no, there is no protein in this substance. Now for the fat, it's called the fat emulsification test. So if you get a cloudy color, then fat is present. If you still get a clear color, then fat is not present in the substance. So that's all you need to know about the fat test. It's called the fat emulsification test. And if you get a cloudy color, then fat is indeed present. And if it's clear, then there is no fat. Our last test, which is the starch, which we are already familiar with from chapter 6, the plants. Now the starch, you're going to use for it something called the iodine. So the iodine solution is a substance that can show if there is starch in, for example, a leaf or starch present in any substance. So you can also use that in the experiment that I explained in the independent variable. You can use the iodine test to show if there is still starch after the amylase, because as you know, amylase is supposed to break down the starch. So you can use the iodine in this experiment. Now back to what we were talking about. Iodine is the solution, as I said, for the starch. Now, what should you get if it's a positive test? What should you get if it's a negative test? If it's a positive test, you should get a blue-black color. You should write blue-black, both of the colors. Don't just write blue or don't just write black alone. No, you have to write blue-black, just like that. Blue-black. If you get a negative test and there's no starch, then it would remain orange-brown. You should write orange-brown. Don't just write orange alone and don't just write brown alone. You should write orange, brown. And for the positive test, you're going to get blue, black if there is starch present. Now, for the food test for the vitamin C, it's called the DCPIP. This is the substance that you're going to be using to indicate the presence of vitamin C in anything. So, what happens if we get a positive result? It will change from blue to colorless. That means it doesn't have any colors. So that is if there is vitamin C in the substance. Now, what if there is no vitamin C? It will stay blue and it will not change color. Then this is the negative result, and this shows that there is no vitamin C in the substance that you are testing or whatever you're testing. Now, this is the last part before we're going to start solving some questions to tackle what we learned today. Now, safety is just a simple part, but it plays a crucial role in like when you're planning an investigation in the exam. So, safety, you should know the difference between safety equipment and safety hazards. I'm going to be explaining what does each of these mean. Now, the safety equipment is the things that you're going to be using in order to be safe when you're doing the experiment, or when if you're faced with some of the safety hazards that I'm going to be explaining now, you're going to be safe, or you're going to be minimizing the like injuries or whatever danger you're going to be facing in the lab. Now, safety equipment, I know some major ones that are very important and are commonly found in paper 6 exams. These are the lab coats, goggles, and gloves. This is like the most important safety equipment that you will be finding in the paper 6 exams. Lab coats, of course, we know what are the lab coats. It's like the white coats that we are wearing in order to prevent spillage of any chemicals into our clothes and to prevent staining of our clothes. Goggles to prevent any spillage entering into our eyes. And these are like plastic, like glasses that are very large in order to protect our eyes. And gloves, of course, we know the gloves that are like maybe silicone or whatever. So I'm going to get you some pictures of them now so you can be familiarized with how they look like. Now, these are pictures of the lab coat. This is the lab coat. It's white in color. And this is the goggles that you're going to be using in order to prevent the spillage of chemicals into your eye. And this is the gloves. And these are just if you're going to be working in a lab, so you can know what are each of the equipment called and how they look like. So the safety hazards is the danger you can be facing when you are working with stuff in the lab. So, for example, you can be prone to chemical burns if you're going to be using harsh chemicals. So, you have to be using gloves and lab coat in order to prevent chemical burns in your skin, maybe spillage of chemicals into your eye and causing some eye damage, and so on. So this is the safety hazards, basically the dangers, and the safety equipment is the stuff you're going to be using in order to protect yourself from the safety hazards. So that's the difference between the two words. Moving on to the improvements to the experiment. Now, for the improvements, for example, if you find an experiment that is written down in the exam and you notice something that is wrong, or you notice that something is done incorrectly, now how do you know them? You're going, for example, I'm going to tell you some of the errors that are done. For example, they use water bath instead of the thermostatically controlled water bath, which means the temperature is not kept constant. So this is also a source of error. And one improvement, you should write down use thermostatically controlled water bath. So this is the improvement to the temperature not being constant. Also, another source of error is maybe not maintaining the pH. So you're going to maintain the pH using something called the pH buffer. This is a device that is used to control and maintain the pH. Also, another mistake that I've seen is using the same test tube or using the same like equipment every time, for example. So if, for example, if they're using the same like if they're using the same measuring cylinder, then this is called contamination. This is a source of error. So how do we improve this? We're going to either say use separate equipment, or you're going to wash them down. Use separate equipment or wash them. So you either wash them or use separate equipment. So of course they might say they didn't wash them, or they used the same maybe glass rod for mixing each of the different test tubes. So this is called contamination, source of error. And how do we fix it? Again, you're going to use separate equipment or wash the equipment every time if you don't want to use other separate equipment. Another source of error is using your eye to judge like the color of something. So of course, everyone has different eyes, and like not everyone can see the same thing. So of course, there's some variation in that. So how do we fix that? We're going to use something called the colorimeter. This is a device that tells you the color of something, and you can use it in like anything that you're testing, and it will tell you the color of something without using your eyes. So for example, some people might have color blindness, some people might have diseases in their eyes, so they might tell you the color incorrectly, or the colors are not accurate enough. So that's what we're going to use in order to measure. And what do you write? We're going to write as a source of error, we're going to write it's difficult to judge the end point. That means that it's difficult to really know what was the color in the experiment, as this person told us like maybe an inaccurate color. So it's difficult to know what really was the color in the experiment. So the colorimeter ensures that we have accurate color measurements. And also another source of error that I've seen is not using the same volume of maybe solution, or not using the same volume of substrate or whatever you're testing. So what is the improvement that we can do to this? We can use measuring cylinder to measure really the volume and to use the same volume in each of the test tubes without getting an unfair calculation. So measuring cylinder to measure the volume of something and make sure that something is constant and the volume is constant. Also, another error that I've seen is not repeating. When you don't repeat and take average, then maybe you got anomalous results in the middle, or you got some inaccuracy and you want to remove the anomalous result, so you have to repeat and take average. Some experiments don't repeat and just rely on the first time that they have done the experiment. Also, repeating and taking average is something very, very, very important to write in your experiment when you are designing the experiment. So again, these are the sources of error that we have said now. You should always maintain the volume of something that you are testing in the independent, and you should use separate equipment, and you should also maintain the pH if you require, and also use the colorimeter when you are working with colors, and always make sure that you repeat the experiment so you can remove anonymous results and remove inaccurate results, and make sure that your experiment is as accurate as possible. Now, to sum up, this is the most important stuff that you should write always in any experiment that you are writing down in the exam. You should always write down the independent variable. You should always write down the independent variable. And always remember that there is no experiment that doesn't have independent and dependent variables. As what is the point of an experiment if you don't have something that you are measuring or something that you are changing? So you should always write these down. You should also write down the constant variable because you need the experiment to be fair and accurate. You should also write down the method that you are using. Now I'm going to be solving some questions right now to show you how do we write this stuff down and there is always a different method for different experiments, so there is no like one specific method for the questions. And you should always include these two things. You should always include safety equipment, and should always include the sentence repeat and take average. These three things will give you 2 marks. If you write down any safety equipment, you're going to get 1 mark, but sometimes in the mark schemes they don't, but like most of the time, just write it down in case if the mark scheme actually gives marks for it. And repeating and taking average, repeat 3 times. This is like preferred. Repeat 3 times and take average in order to make the experiment, as I said, very accurate and free of anomalous results. So this is just like the most important stuff that you should include in any experiment that you write down in the exam. Now, this is the first question in the, I'm going to be solving some questions with you guys to tell you how to write it down and how to write down the experiment and what you're going to be awarded for in your answers. So, after pollination and fertilization, seeds will develop. These will disperse into new environments. If conditions are suitable, they will germinate. A student put some seeds on wet cotton wool in a petri dish and left them to germinate. The student counted the number of seeds that had germinated. Describe a method the student can use to find the best or optimum temperature for the germination of seeds. Now, we're going to just first of all identify the independent and dependent variable in this experiment. So I've noticed right now that the independent is the temperature, as they're trying to find the optimum. So of course they're going to be using different values of temperature to know like how it will affect the germination of seeds. So temperature is the independent variable, and notice here they didn't say the effect of. So this is why I'm telling you guys, you should understand the experiment before like doing the effect of thing that I've told you about. So here they didn't say it, and you should understand the experiment. So they're trying to find the optimum temperature. The temperature is the thing that they're going to change constantly to try to find what is the highest rate of germination of seeds that happened during this temperature. So this is what's meant by the optimum temperature. So this is our independent. And of course, the dependent is going to be the germination of seeds, but of course, we're not going to write it down like that. We're going to write the number of seeds that germinated. This is the dependent variable. We're measuring the number of seeds that germinated during or in this specific temperature that we're trying to find. So, first of all, when writing down the experiment, you can just write it down like that. You can write down, use at least 3 different temperatures, but I like to organize it like that. I like to first write it down like that. I write it down like that in the exam. Independent variable, and you put down 2 dots like that and say that it is the use at least 3 different temperatures. Now, I'm not going to write down the words fully because I need to save time to explain. So you should write down, always write down the words. Like, don't write it down like this. You should write it down like this. Don't write like abbreviations or incomplete words. You should always write down the words fully. So use 3 different temperatures, and you should include some units. You should include, maybe say, use temperature 50, maybe 37, and 10, for example. Now, this is the first mark that you're going to get, the independent variable. Now, you're going to also say that the dependent variable, maybe you can skip the step of writing down the independent and dependent variable, but this is if you get like confused and you're disorganized. So you can do it like that, independent variable and dependent variable and so on. Dependent variable over here is the number of seeds that germinated. So always notice and always make sure that the dependent variable is something you can physically measure, and it's just not like maybe germination of seeds. Like, what do you mean by germination of seeds? Like, what exactly is the germination of seeds? So you're going to say the number of seeds germinated. So that's our dependent variable. Now you can write down the constant variable in this experiment. It is, of course, the pH value. You can maintain using pH buffer, as I explained earlier. You can also maintain, let's say, the species of the seeds. Like, you can't use different species of seeds. So make sure all the species are the same and they are like one species, so you can make this test a fair and accurate test. Also, make sure that they are the same age, because at different ages, the germination of seeds will be affected, as we all know. So you should get the same species, the same age, and yeah, these are the 3 constant variables that I can think of. So this is the, let's say, the 4th mark or something. So now for the method itself, how we're going to do this experiment, we're going to write down first of all, you're going to have to also maintain, sorry, I forgot to write that down. You should also maintain the water concentration, like the water levels, of course, because if you give it more water, then it will maybe affect the rate. So water levels. Now for the method, you should use at least 10 seeds in each of the maybe dishes that you're going to be putting in different temperatures. Now, for the temperature, you can write down that we can maintain the temperatures using the thermostatically controlled water bath that I showed you earlier. Just give me a moment, I'll write it down. So you should also write down that you should use at least 10 seeds, because if you use one seed, then this is like not a accurate example or accurate test. So you should use at least 10 seeds in every dish that has different temperatures. So use at least 10 seeds in each dish of different temperature. Okay. So you're going to also say that the optimum temperature, or the temperature that has the most seeds that germinated, then that means that this is the optimum temperature. So let's say maybe 8 out of 10 of the seeds germinated in this temperature, then this is the optimum temperature, or the best temperature for this seed to germinate. So you should also write this down. You should write down that um, check how much seeds germinated in each temperature, and the higher the amount of higher amount of seeds, sorry, that germinated, then this is indeed the optimum temperature. So in each temperature, and the optimum will have the highest seeds germinated. So this was for the method. Now for the safety, you can write down use maybe lab goggles, lab coat, and gloves. And you can also say you can repeat each of the dish 3 times and take average of the temperature. Repeat each dish, or let's say each temperature, 3 times and take average. So you should always write these points down. You should always like put these 2 points down to like guarantee some marks. Sorry. Okay, this was the first experiment we have now. So if you want to make sure that you were correct, you can open the mark scheme for the exam that you're solving and check if you have written down everything that they have said. Now for question 2, a student wanted to find out if starch was present in both leaves grown in direct sunlight and leaves grown in the shade. Plan an investigation to determine if starch is present in both types of leaf. Now from reading, I've noticed that we're going to be using the iodine food test, because they're trying to see if starch is present or not. So now we're going to be identifying the independent and dependent variables before we start writing the experiment down. The independent variable is whether or not the plant was given direct sunlight or it was in the shade. So we're going to be saying the presence of light, or the amount of light given to the plant. And the dependent variable is going to be the presence of starch in the leaf using the iodine test. So let's start writing down the experiment. The independent variable, the thing that we constantly change, is the amount of light given to the plant. Now, of course, as I said, you can skip this step by like writing the independent is this and the dependent variable is that. You can skip that step and you can just go on and writing the experiment, but I'm going to be doing it just in case if you get confused. Independent is the amount of light given. Now the dependent variable is going to be the presence of starch using iodine. Now, for the constant variables, of course, we have to keep some stuff constant over here. Now, we can't use different species of the plant or different age of the plant. Now, if you're going to use different age of the plant, like plants that are maybe one plant is 1 year old, let's say, and the other plant is like 9 months old, you can't do that. All the plants have to be the same age and the same species, as the species can be affected. Maybe you're using a variegated leaf that has some white spots. So you have to make sure that all the plants have the same species, same age, and also we can keep the pH constant over here, and we can keep the temperature constant. We can keep the carbon dioxide concentration constant over here. So there's many things we can keep down constant over here. So I'm going to be writing some of them. pH, species of a plant, age, so, and also temperature, because the only thing that we're changing over here is the light intensity. Now for the method, we have to use at least 3 plants that were grown in the direct sunlight and at least 3 grown in the dark, because we can't just measure, as I said, we have to, in the independent variables, we have to use at least 3 values to keep the experiment as accurate as possible. That's because we never know, one plant maybe has some problems, the other plant, whatever. So we have to be using 3 plants at least. So we're going to be telling them that. Now for the first thing in the method, I'm going to be saying use 3 plants from grown in the shade and direct sunlight. Now the next step, if you are already familiar from chapter 6, we should remove the chlorophyll substance first before testing with iodine. Why? That's because the leaf is already green and it will have green substance that will react with the orange-brown iodine color, and it will form you a blue-black color. That's not because the starch is present, that's because it's already green, and green with the orange-brown will form like a blue-black color alone. So we have to remove this substance, the chlorophyll substance, and we have to also remove the cell membrane, because and the cell wall and all of this stuff, so we can allow the iodine to penetrate into the leaf and not keep any barriers from the iodine going into the leaf. So we have to first boil the leaf to remove the cell membrane. And we're going to be adding the ethanol to actually remove the chlorophyll substance and to allow an accurate measurement of whether or not the starch is present. So we're going to be rubbing with ethanol the leaf. To remove the chlorophyll substance. Now, we have to also, when we remove the cell membrane and we remove the chlorophyll, now we're going to rinse the leaf to keep it like smooth. And now after all of this stuff, we boiled the leaf and removed the chlorophyll, we rubbed it with ethanol. Now of course it's going to be dry and brittle, so we have to keep it smooth again by rinsing it in hot water again to keep it like moist and not cracked and dry and brittle. So we're going to be putting it in boiling water again. Now we're going to be using the leaf, we're going to be putting it on a white tile. The white tile is something that's like a normal white tile, and you're going to be putting the leaf on it and adding the iodine to indicate the presence of the starch. Now, why do we use the white tile? That's because it is clearer, or it's clear whether or not the iodine went into the leaf, and it's clear how the leaf looks like. So it just makes everything like more clear to see. So we're going to be saying put the leaf on a white tile and add the iodine solution. And of course, whenever we talk about the food test, we have to say the positive results to gain more marks. So over here, we're going to be saying we have to, if we get a positive result, then it's going to be blue-black in color. Now, that's basically it. You have to first rinse the leaf, and we're going to be adding the boiling water and the ethanol because, as I explained, we have to remove the green substance at the cell membrane and the cell wall to allow the iodine to enter into the leaf. Now, the last thing, as I told you, in any experiment that you're planning or writing down, you have to write the safety and you have to write down the repeating to gain more accurate results. So we're going to be writing the safety over here. We can write that we should use gloves because we're dealing with some of the iodine, which is, of course, a chemical substance that may cause some irritation in the skin. Use gloves and maybe wear a lab coat and goggles. Okay, so this was everything that I wanted to say to you today. Please consider subscribing to my channel and like this video if you enjoyed it, and share it with your friends if you have learned something new. So please, if you have any suggestions or any feedback, please leave it in the comments. And if you want me to solve for you any specific paper or a specific variant, then please leave a comment and I'll reply to you as soon as possible.